Well, hi, I think we're recording. I'm going to have to make sure when we get done. This is the second video that's going to be on uh, probability that's used in the aspects of doing genetic problem solving, understanding how Mendel's laws work, etc. I may be glancing to my left a little bit. That's because that's the screen that you would literally be looking at, just to make sure that we're on the same page and things are working. So let's continue on and see then what's happening. Keep in mind what we're really talking about here is probability is the occurrence that something can randomly happen. And I know for many people this idea of random events is a difficult one to think about because how in the world would God use random events? He's creator, he has his plan, and you hear a lot about these things. So nothing is random. Well, that may be too simplistic an understanding of what we mean by the term random and we mean by how God is in control, etc. Because I don't think any of us think that we are being puppeted by God and he's holding us and moving us around and we do only the thing that he tells us to do and we have no free will. Yet we throw out some of these terms. So let's just try to get it for today when we start to look at what do we mean by random. So the idea is, can we start to really make predictions based upon Mendel's laws of segregation and his law of assortment? Can we try to determine what could happen, and if it could happen, how do we help to verify that those results can meet our predictions? So that's kind of the purpose of understanding today and on this video. So when we do so, let's kind of look at today what we mean by this aspect of having this probability. But we're going to watch a video to help us here. Oh, have to go back, try it again. It's always some of the problems with clicking here. This is Bozeman Science. See what you think.
Hi, it's Mr. Anderson. Today I'm going to be talking about multiplication and addition. You might think that's a little simple for what I usually talk about, but I'm going to talk about how we can apply multiplication and addition to genetics. And unlike in math, I'm going to start with multiplication because it's actually easier that way. Multiplication is going to occur in genetics if we ever have independent events that are going to occur in sequence. Another way to tip yourself off to that is if in the question it's saying the word and. So let me give you an example of that. What are the odds of flipping a coin 5 times and getting tails on every flip? Well, you can use the rule of multiplication to figure that out. So what are the odds of getting a tails on one flip of the coin? That's just 1 and 2. So all you do is you multiply all the probabilities out. 1, 2, and that's going to be 1 and 4, 8, 16. So that's a 1 in 32 probability of flipping the coin 5 times in a row and getting tails every time. Same thing would be true if we were to say you're going to have 5 kids. What are the odds of all of them being boys? It's just multiplying the probability of each of those events because they have to occur in a specific sequence. So let me show you how we could apply that to a couple math and then genetic problems. And so you may want to, as you watch the video, pause the video after I present the questions, try to work them on your own, and then I'll go over the right answers. So here's the first two. Number 1, rolling snake eyes on 2 pair of dice. So how do we do that? Well, there are 6 sides to a dice. A 1 is going to occur on 1 out of every 6 of those sides. And so it's a 1 in 6 probability of rolling a 1 on 1 dice times a 1 in 6 probability on the other dice. And so the right answer would be 1 in 36. So it's going to be rare that you get snake eyes, roughly 3% of the time, less than 3% of the time. Let's go to the next one. What are the odds of picking a 3 out of the deck, returning it to the deck, and then picking a king out of the deck? How do you solve this one? Well, you have to look at each of those different events because they're happening independently of one another. What are the odds of picking a 3 out of a deck? Well, there are 4 3s in a deck of normal playing cards. There are 52. And so to save myself a little bit of time, I'm going to reduce that. So that's 1 in 13 probability of picking out a 3. Since you're returning it to the deck, there are going to be 52 cards again, so the odds of getting a king out of the deck are also going to be 1 in 13. And so the odds of doing both of those in succession is going to be 1 in 169. So it's going to be really, really rare. Okay, so that's math. Now let's apply it to genetics. Let's apply it to the work of Mendel. So in this first problem, in a cross between pea plants that are heterozygous, so they're hybrid for purple flower, what's the probability that the offspring will be homozygous recessive? Okay, so how do we do that? Well, we first write each of the parents, so big P, little p, crossed with big P, little p. So what are the odds that this parent over here, since we're looking for a homozygous recessive, to build that, we have to get a little p, little p. So what are the odds that this parent is going to contribute that little p? Well, that's a 1 in 2 probability. What are the odds that this other parent is also going to give a little p? Well, that's a 1 in 2. And in order to get that homozygous recessive, we have to have both those occurring. So we simply multiply those, or that's going to be a 1 in 4 probability. Now,
Well, as you can see from Paul Anderson's introduction, it really does apply to genetics. And what you may notice that he never did use a Punnett square, which a lot of you have used in the past. And that's part of what we're going to try to accomplish here. Now you can see the rest of that video that Paul Anderson does just by going to bozemanscience.org. I'll give it to you in class to make sure. Or just look up Bozeman Science on YouTube and it'll come up in his channel, and you can look for his ones on probability and statistics, and it'll be very helpful. Anyway, so let's kind of continue what we're on here. So what we're looking now, if you go into your course notes, we're trying to get an understanding what this probability and statistics actually mean. So as you can see from your notes, we're defining it for you. What is the probability? And please notice it's an italicized P. We're going to get seeing lots of P's, but when you see the italicized P, it's the probability that something would happen. What's the probability that an event would occur in the number of times the event does occur? So in other words, you divide it. So what's the probability of rolling a 3 with a single-sided die? Well, as we saw in the video, that would be 1 out of 6. So it's at 16.6%. What's the probability of the next child born in the U.S. and North European descent would have PKU? Well, knowing that, what would be the choice? You have to kind of look at what the data says. And so when they look at the data, they give you a probability of somewhere between 1 in 10,000 or 1 out of 15,000. So that's from statistical data that they have acquired, and they look at the occurrences, and when they've looked at the data, that's the chance, that's what actually occurred over a series of time, and so they put it into a fractional form as well. All right, so let's kind of then look at example 3. What's the probability that an offspring from a self-fertilized monohybrid will show the same dominant phenotype? So you have this parent cross. Now notice that P here is not italicized. That P is actually a standard one. So that refers to the parent generation. So if you have a homozygous large A or capital A being crossed with a homozygous small a or little a, then the chance of it coming up to having the same kind of dominant phenotype, what we're looking at it has to have at least one big A. Well, how you solve it by using probability rules is you would have a chance of this aspect where when you cross this, when you have a self-crossed hybrid, what you end up getting is a 1/4 chance of it being big A, and a 1/2 chance of it being big A, little a, and a 1/4 chance of little a, which then really means you have a 3/4 chance of actually having that phenotype. Well, 3 out of 4 is a 75% chance, or 0.75. Well, that's what we refer to as bringing in this probability and this statistic. So what happens when you want one situation or another situation? Now, what we are referring to is one of these are mutually exclusive. It either will happen this way or it will happen that way. It's not they can both happen at the same time. You use what is known as the sum rule in probability. So the probability of one event happening out of 2 mutually exclusive events is actually the sum of their individual probabilities. So let's look at an example. What's the probability of a smooth yellow or a wrinkled yellow pea that comes from a plant which is self-fertilizing of a dihybrid. Now don't forget, you can always pause these videos and think and process and then write things down. You don't have to just keep playing. So let's just take, for example, what we're being asked here. What's the probability of a smooth yellow or a wrinkled yellow pea that's coming from a self-fertilized dihybrid? In other words, a big S little s, big Y little y, being mated with itself, big S little s, big Y little y. You do that a lot with plants, and you'll get to experience that when you're looking at your mustard seeds, which you, I think, germinated yesterday. So if we look at this, you'll start to see what's the probability of having one big S and one big Y, okay, and that's 3/4 times 3/4, and we can actually go back and look at that from the previous one. So let me just go back and show you what I'm referring to. Remember, we came up with here 3/4 chance of having, if you cross the big A, little a, showing the dominant trait. So let me go back to where we just were. So what's the chance of showing a big S is 3/4. What's the chance of showing a big Y is 3/4. So it's a 9/16 chance. What's the chance of it showing two little s's was 1/4. And again, what's the chance of seeing the big Y is 3/4. So it's a 3/16 chance for that event. But because we've asked what's the chance of this or that, then you literally add those two probabilities together. So you get 9/16 plus 3/16, 12/16, or now it can be reduced to as 3/4. So you need to kind of practice this one. So let's practice this. You can do this with a colleague, you can do it with somebody else up in the dorm, you can try it on your own, but try to practice it and see how well you do. Run through either one of the two types that we just went before and then see how well you do. Then actually start this rationale. Why would it be the sum rule? When do I use it? This whole idea of don't just write things down, but process what is it and why is it and how is it, instead of just writing information down and thinking hopefully you memorized it that way. All right, so let's look at the second probability rule. This probability rule is called the product rule. Now, what's happening here is we're looking at two independent events occurring simultaneously. Two independent, they're not tied to each other, they're independent to each other. And what happens there is we're looking at the products of these individual probabilities. So let's take an example, so hopefully that will make sense to you. A couple who are both carriers of the albinism mutation. Okay, so if we look at, for example, big A, little a, and we think that albinism is a recessive trait, so we'll say it's the little a, we want to know the chance of having an affected child. So we can start to use the probability aspects in the product rule. The probability of getting a little a from the mother, okay, of this couple, thinking that if they're both big A's and little a's, the chance of getting the little a would be a 1/2 probability. The probability of getting a little a from the dad, if we had two that way, would be a 1/2 chance. So then using the product rule, both of these simultaneous events have to occur at the same time. You multiply those separate probabilities. Therefore, the chance of having little a from both parents is actually a 1/4 chance. Now, again, with these kind of problems, practice. Make sure you understand, when would I use sum rule? When would I use product rule? Why would I be using it? How do I incorporate it? What do I need to know? And if you have any questions, always bring them into the beginning of the class after doing a flipped portion. Okay. Well, what about pedigrees? Let's kind of look at some pedigrees and see what happens. We are going to apply those kind of probability rules as we solve these. So let's take a look at a pedigree right here. If we start to look at these pedigrees and we want to look at information on this pedigree, we're going to start to look and see that certain things are going to be happening at specific times and what's actually transpiring. So let's kind of, what do we see actually here? So let's just start off. Let's assume that this is an individual who has a little a up in the top and a big A on the right. Okay, so little a on the left because it's an affected child, big A on the right. And how would we know that it would be a big A? Well, we don't know, but we always make that assumption unless there's some detail given to us that can help us to know better. We talk about the parents as being number one, the parental generation. So it'd be like 1 would be the little a, and 1 would be the big A. So if this kind of makes sense to you right here, that would be 1, and this would be 1. If we go to level 2 and we start here, this would be 2, 2. This would be 2, and this would be 2, because it's all from that second generation, would be the first filial. But would you agree that what we see here is that this parent, when crossed with this parent, would produce that kind of child? And if we cross that with here, big A, the question is, what would this child be down here? So what would 3, 1 be? What would 3, 2 be? And what would 4, number 1 be? And I don't think that we use this symbol because we don't care necessarily what gender it is. We just want to know if the child would be affected. Now we look at these two and we would say on 2, 3, and 2, 4, how do we know that these would be big A and big A little a is because we look down here and we have a affected child that came from parents who were unaffected. The only way to be affected is to have a little a from both parents, but the only way for both parents to actually not show the defect would be that they would have a dominant gene. So these clear areas tell you that each parent has to have a big A. This child tells you that the other gene in here has to be a little a, or you couldn't have had this child. Now, knowing that, we take a look at these two children and we have to kind of figure out what's the probability of having a certain kind of offspring. So if we continue this a little bit further, we start to ask, let's see if it makes sense to you. What's the probability that P3, 1 is a carrier? So if we come over here and we look at P3, let me just back up a second. So what we're trying to solve is, what are the P3, 1, and 2 probabilities? What's the P4? That's where we have these blanks. So what's the chance that this one's a carrier? Well, we look up it here and we always make the assumption that anybody coming in is homozygous unless for some chance we actually know something below that it would not be. This would be one way to tell you it wouldn't be, or we would actually say we know that that person was coming in, but we don't. So we make the assumption big A, here's big A, little a, we know from this parent. So what's the chance that this would be a big A, little a? Well, this one would have to give the little a as well, and that's a half chance. So there's a half chance of being big A, little a there. Okay. What's the chance that 3, 2 would be a little a? When we look at this one, and I look at carrier, the chance that this would be a big A, little a, the carrier chance of this one being a big A, little a is, well, this parent and this parent, we know are that. We know that it has to have a big A, but what's the chance that would have a little a? Well, as we talked about maybe back in freshman year, you may remember, but if not, let's just help you. The answer for this is 2/3. And why is it 2/3? Well, the reason it's 2/3 is because I know that it has a big A. When you did a Punnett square, when you do this cross, to have a big A in here and have a little a as my second option would be 2 of the 3 possibilities. So if we want to think of it this way, when you cross a big A, little a with a big A, little a, 2 of your 3 choices of your cross end up having a big A, little a. So let me see if I can kind of remind you by using, there we go, there's the pen, a little aspect here. So if we would have done a Punnett square, let's just do quickly a Punnett square. Sometimes it helps to visualize it. And we knew that this person over here could give a big A and a little a. And this person could give a big A. Oop, see, that's one of the problems I have with the program, is it sometimes does not help me on that. So let me come back here and try this again. Let's go back and up. So there we are again. So let's continue through. The idea that I was trying to get to, maybe we can see better in the next one, you end up having 1/3 chance. So let's just take a look. How do you calculate probability? So let's come back here. Let's use this. Let's make this a big A and little a, and this a big A and this a little a. Then what you'll notice is that this one has 2 big A's. This has a big A, little a. This has a big A, little a. And when you end up there with 2 of them have big A, little a's. So that's why this one ends up having a 2/3 chance of being a big A, little a. Now, knowing that, if this was a big A, little a, and this was a big A, little a, it would be a 1/4 chance of having 2 little a's down here. Well, then I just have to say, but I'm only half sure that it has big A, little a. I'm only 2/3 sure it's big A, little a there. So 1/2 times 1/2 times the 1/4 chance tells me the chance is a 1/12. Now we'll work through this in class, but pause, see if you make sure you understand, and then at that point what we'll actually start to look at is there a way in which we can practice this and have a good grasp of it. Okay, let's continue on. The third kind of actually is starting to look at what's the potential outcomes that you are looking at for calculating any kind of specific probability of some specific set. So here again is a standard Punnett square that you've dealt with. The P's are typically the dominant traits, the Q's are the little recessive traits. So if I have a heterozygote crossed with a heterozygote, I get 2 big A's or 2 big P's. I can make that P square. This becomes PQ, that becomes PQ, and this becomes Q square. In other words, it's big A in the upper left box. It's big A, little a, and it's 2 little a's here. That's how you get to that kind of expansion. Well, this all comes from multiplying P plus Q. So if this makes sense, the number of P's and the number of Q's is equal to 100%. Okay, well that's kind of obvious. The number of P's to the number of Q's is equal to 100%. So you can't have any more than 100%. So P plus Q square, or P plus Q, would end up coming up with these results. That's the number of trials you want to do. Okay, let's just take a quick pause and realize we're going to practice this in class. So make sure, go back through, write down your information, follow along in your reading, and when you finally get to the end, write down some questions you want to bring into the classroom time to start the day off. And then we'll move on from there with how to use or work with this information. Let's just see how we do, and let's give me any answers or questions that you have to anything that when we see you tomorrow. Okay, you guys take care. See you at 8 o'clock.